Okay, so part 2 of online lecture number 9, I suppose. So when we work with microbial cells, we talk about their requirements. A few liters of the lab you can make in a few liters of the growth medium you can prepare and sterilize in the lab. But when you're working with industrial processes, industrial synthesis, manufacturing plants, thousands of liters that you cannot make in your lab. I can tell you from my personal experience, when I was doing my master thesis, I used to have around 120 liters of growth medium every week. That's a lot for a student to prepare. But when you're working in industry and you're working for synthesis of products like insulin or like I said, interferon or some other therapeutic compounds, that's nothing. You are going to be literally working with thousands of liters, and for that reason, you need an industrially set up system for the synthesis and the sterilization of this growth medium. As discussed a number of times, a growth medium in which the microorganisms can thrive, it must have their necessary ingredients, and it depends on the type of the cell, the final purpose of the cell, as well as the byproducts. The byproducts are important because when bacteria thrive, they would have to have their excretory byproducts. And the byproducts themselves can choke them, can poison them. And when the byproducts levels and concentrations increases, it could make the other bacteria halt their thriving, their production, their synthesis, their reproduction, their division. And the final purpose, what is the final purpose of the cell culture? You want to have some investigations of a drug, you want to have a recombinant protein produced, a vaccine. So it depends from application to application and the cell culture to cell culture. And according to your situation, you will design your growth medium and the type of cell as well. Very important. For example, some cells would like to grow under pH 7, some would like to grow under pH 4. Similarly, some cells, some bacterial cells, in this case, some fungal cells or aspergillus, other candida cells, they would like to grow in the presence of sucrose, the temperature of 37 or salinity of 8 pH. So it all depends on the specific type of cell and purpose, and then you can design your growth medium for them. Yeah, it's already discussed. You can grow them in batch cultures. You don't need any dilution. Batch culture means like you have a big culture. You have all the growth medium supplied to the culture vessel where you are going to conduct your cells to grow and thrive. And then you wait for the highest density to reach. And then eventually in their log phase, they will have their maximum density reached. And you can limit the growth by nutrient availability. So at the end of the exponential growth, there will be nothing left. And eventually the batch cultures you can harvest and you can centrifuge, you can have the cells if you're looking for the product inside the cells, or if the product is released into the medium, then you can have the medium as well. So you can use them for batch cultures. This is the good thing about bacteria and yeasts. Okay, this is something the nutrient medium we had already discussed in great detail, so I will skip over the slide. Now, the types of growth medium, there are two common types of growth medium. One is known as complex medium and the other is known as defined medium. Media is actually the plural. So when I say medium, I'm referring to singular. Media is plural. Complex media has one or more than one crude sources of nutrients, and we do not know their exact composition and we do not know exactly what is inside the exact proportion of these chemical constituents, but we know that it has these XYZ ingredients and it really works very well for routine cultures. This would be like if I say, bring me bring me fructose. You can go and you can get me a mango, which is a lot of fructose. You could squeeze the pulp or the juice from the mango, and there's definitely fructose. We know about it, there's fructose in it, but we do not know exactly how much fructose it is. But if I tell you, bring me a solution of fructose 5 milligram per liter, then we know we have to have 5 milligram and we have to dissolve it in 1 liter. So that's exactly defined medium. The complex medium and defined medium, there's the difference. We know there's fructose and we know the exact amount of fructose. It makes it defined. In the case of complex medium, we know there's fructose, but we don't know how much fructose is there. So the defined medium is a synthetic media containing chemical compounds, and we know the constituents, each and everything, because we make it. We do not harvest it from some natural sources. For example, complex media in case of mammalian cell cultures or in case of bacterial cell cultures, we have an important compound, we call it yeast extract. Now the yeast extract, we do not know exact constituent compositions, but we know it is a lot of organic sources. It has a lot of organic amino acids. But in case of defined medium, we have the synthetic medium. It contains everything. And we know about the exact proportion, what is what and in how much quantities. Now, we're talking about mammalian cell cultures. Animal cells or any mammal cells, we can harvest, even tissues, even organs can be removed from an organism and they could be cultured artificially, just like bacterial culture and fungal cultures and plant cultures. In a plant culture, which we discussed in the last chapter, we said any part of the plant can be cut from it, excised from it, and that part is known as explant, and we can place it under defined medium and they will grow. They are totipotent, they can transform into a whole new plant. But in case of mammalian cells, this is not going to be the case because the mammalian cells, they are not totipotent. So if you culture a keratinocyte cell, it will always grow and make you keratinocyte cell. It cannot give you a whole organism, a whole rat or a whole mouse or a whole elephant, no. Because these cells are monopotent, they are not totipotent. The mammalian cell culture allows us to investigate, to capture the activities, for example, the control of the cell cycle, for example, the effect of a number of genes. Reverse genetics was also one of the topic in the last lecture. Somebody asked me, you can also do reverse genetics. So reverse genetics is a complex field of genetics in which you go for detection of genes from phenotype. So you do not go from genotype to phenotype, but you go from phenotype to genotype. That's the only difference. You first get the proteins, and from the proteins you go towards the gene. You want to identify the genetic makeup from the proteins. Okay, cell culture and media reagents, but I think I need to stop.